We're going to cover a lot of information, and we plan to answer your questions and share your comments at the end. Our Q&A session will be divided into two parts. We'll call upon individuals who've raised their hands over the first 15 minutes or so, and then we'll switch to the written Q&A to allow those who would prefer to submit their questions in writing to have a voice. On your screen at the bottom, you'll see verbal Q&A, raise your hand, written Q&A, use the Q&A box. If you just want to give a comment, feel free to do that using the chat feature. And if by chance we have too many questions in the time allotted for today's meeting, we'll make sure to follow up directly with those individuals via email in the next 7 to 10 days. So just to give everybody context about the site that we're talking about, where this building is situated or will be situated, we are located in Southwest Berkeley, adjacent to Aquatic Park and the railroad track that runs right behind our property. We're very happy to be longtime business residents in Southwest Berkeley. So our agenda for this evening and our speakers, Jens Vogel, who I'll introduce in a moment, is our Senior Vice President and Head of Global Biotechnology for Bayer. Drew Johnston is our Vice President of Site Engineering here in Berkeley. And my name is Kathy Keck. I'm the Interim Head of Communications, and I'm tonight's facilitator. And with that, I'm going to turn the program over to Jens.
Thanks very much, Cassie. And first of all, very warm welcome from my side. And Cassie, thanks for emphasizing the senior. I know I'm graying, but no, a very warm welcome. I'm excited to be here tonight. Just to introduce myself, so I'm the Global Head of Biotech, and I came here to Berkeley and the Bay Area 20 years ago, and really fell in love with the city of Berkeley and the community. It's a very special place, so I feel very much part of this community, and this is home for me, as it is for many of you. So let's maybe go to the first slide, and I will show you and tell you a little bit more about who we are as Bayer Berkeley and what we do. So I cannot see the slide yet, but it's coming up. There we go. And yeah, so first maybe, really about who we are. We are the global headquarters of our biotechnology organization, and we are really a very diverse team of over 1,000 people. You can see here that, and this is part of what I love about our community, because it very much reflects our community, what you see here. We have about 70% people of color, and this diversity in every shape or form, in terms of racial diversity, gender diversity, diversity of thought, is really what, to me, is directly linked to innovation. And that's something that we can do better here, I believe, than anywhere else in the world. You can see that we have also very experienced teams. And you can also see something that we're also very proud of. We have all levels of education, because we are a manufacturing site, a development and manufacturing site. We have high-level scientists, PhDs, masters, bachelors, but also really a lot of people all across all levels of education. And this is also reflected in the jobs that we want to create going forward. And we talk about that a bit today, because we have really big plans in also expanding our presence. Next slide. So we've been here for over 30 years, and what we really do is we make medicines and really life-saving therapies. That's why we come to work every day, and that's why myself and a lot of us really got into this field to begin with. Specifically now for many years, for decades in fact, we made products, biotechnology products to treat hemophilia in particular. So we have several products that we make and then from here distribute globally in over 50 countries in the world. And hemophilia, for those of you who might not know, it's a very devastating disease. It's caused by a gene defect, which means that the patient's blood does not coagulate. If they cut themselves, if they have a wound, they basically don't stop bleeding. And you can imagine that historically, people with these children with these diseases, with this disease, did not have a very high life expectancy. Now, on the right there, you see one of our patients. His name is Evan, and I had really the pleasure to meet him when we celebrated World Hemophilia Day a while ago. It's also where the pictures on the left were taken. And he's a perfectly healthy young man, except he has hemophilia, but that he combats by just taking routinely one of our products. And he can have a perfectly normal life. He's training to be a nurse right in the middle of the pandemic. And this is really why I am in this field and why so many of our people are in this field, because we help people live a normal and full life. And that's what we really draw our satisfaction from and what we're very proud of. Next slide. So what we want to talk about here today is really our next step in our journey, which is we want to go into cell and gene therapies, because we really believe that these therapies will revolutionize medicine. Now, many diseases are actually caused by specific gene defects. And I just showed you one example. I mean, hemophilia is a prime example of that. So these gene defects that are then present in many or all cells and basically then make it very difficult to treat those diseases with traditional medicines, especially chemical drugs. Now, other diseases can be caused by cell damage. For example, Parkinson's disease is actually believed to be caused by neurons, brain cells that normally produce a brain chemical, or dopa, which unfortunately then these cells start to die in patients, causing Parkinson's disease. Other examples are heart failure. There are many things that can cause heart failure, in which then heart cells die ultimately, and ultimately leading then to irreversible heart damage and deaths. So cell and gene therapies really are trying not to just relieve symptoms, but for the very first time with these therapies, we could potentially shift the paradigm to an actual cure of many of these terrible diseases that are afflicting so many of our friends and families. Next slide. So cell therapy specifically means that you actually take, in the case of autologous cell therapies, patients from cells from a particular patient's body. You engineer them to address basically the, for example, gene defect that those cells might have, or you also might train them to basically be more effective at whatever function they actually carry. Then you infuse them back into the patient to treat the underlying disease. Allogeneic cell therapy is what we are after, which is really the next step in cell therapy, in which you basically take cells from a healthy donor and create something like a universal cell bank, which you can then use to really treat many patients on a large scale globally. And this is what we are after here, because the first generation of autologous cell therapies have shown spectacular results. For example, in treating cancer, hematological cancers, we've seen response rates of over 90% in many cases. And so many patients can live for many years and or are indeed potentially cured. So that's really revolutionary, but we believe we need to bring these therapies to more patients. And the autologous approach, if you do it really patient by patient, is extremely difficult, hard, and you cannot bring enough therapy to enough patients on a global scale. That's why we are going into allogeneic cell therapies. And this is also something we want to manufacture right here in Berkeley in this new building. Next slide. The other aspect, again, gene therapy works by treating the disease that is caused by a gene defect by actually delivering the intact gene into the patient's body. And therefore, so in the example of hemophilia, we actually do have an hemophilia gene therapy in the pipeline that we are developing. We hope that ultimately down the road, we are able to repair the gene defect by delivering the intact gene so that people like Evan don't necessarily have to continuously take our medicine, but they're actually cured. So that's the goal that we are after. And we have many therapies in the pipeline now. Parkinson's is actually our first target, and we are hoping that this will be the first cell therapy to be manufactured in this new building here in Berkeley. So a cell therapy that can potentially really cure Parkinson's. Heart disease. And there's a video on my LinkedIn page, for example, that if you're interested in that, which I think is quite impressive, where we are working together with Blue Rock in our research unit on the East Coast on basically not engineering, but culturing heart cells in bioreactors and then transferring them back into patients so we can actually repair a damaged heart. And of course, cancer that afflicts so many people worldwide is something that is very much on our mind with these cell and gene therapies, there in particular with cell therapy. And we have a partnership with Atara, which we recently made public, where we are developing a next generation CAR-T therapy to treat solid tumors in several indications. So that's what this is all about, developing and manufacturing these new therapies and bringing them to more patients so that we can treat these devastating diseases. And with that, I'd like to hand over to Drew to talk more about the building itself.
Thank you, Jens. I appreciate all that. And it's always a nice introduction for me. I've seen that presentation a few times, but it's always fun to hear about what we're actually trying to do here, which is marvelous. So all of that takes facilities, and that's what we're here to talk about. So I'm going to go over the highlights of our cell and gene therapy production center. So this would be the facility where the late phase clinical trials and the first commercial launch goes. Any approval of a facility is on a timeline, right? So maybe you can click through this slide. You'll see we've been at this a while. So back in August of last year, we made our original submission. We received comments from the city in September. We submitted our final application or a resubmittal of our application in November. First community meeting to allow the public to have comments and a live interaction with us was in January. We had a design review, which is part of the city process. So that is a commission through the city. Also a public meeting the end of January. So, and here we are, beginning of March for our second public meeting. Just bear with the community. And then next week on a Thursday evening, we'll meet with the Zoning Adjustments Board, which is also a city approval body to discuss the facility, which is also, again, a public meeting. So maybe we'll see you there. Next slide. So I know in the opening slide, there was a high-level view of the Bear campus where it sits in Berkeley. This is a zoom in on that. 7th Street would be on the right, the bay would be on the left, north is up. You can see on the west side of our site along 4th Street and between 4th Street and the railroad tracks is where most of our manufacturing is. And you can see the cell and gene box there. That's where we plan to put this facility. Next slide. Little different view of our campus now. We have two zoning agreements or use agreements with the city. One is from a development agreement that we that was enacted in 1992, and that's the colorful boxes that are between Dwight and Carlton. South of Carlton down to Grayson, we have a general use permit that governs the development of that piece of our site. You can see that the part that's covered under our development agreement with the city of Berkeley, we have various use parcels that have various land uses, so administration, manufacturing and such, and also various heights. You can see that there's two that are allow up to 80 feet for production facilities, the purple and the green. And again, the cell therapy facility we're talking about is in the green box there. There's also some setback requirements here, but there's a slide later in the deck that kind of highlights those, makes it a little easier for me to understand. Okay, so here's a picture of the facility. This is looking northeast. So if you're standing on the southwest side on the railroad tracks looking up towards Albany, this is the perspective that you'd have. You'd also have to be in a drone. So the facility is around 100,000 square feet, 98,000 to be more exact. It's a mixed manufacturing facility. We have 4 floors in the facility, three full-size floor and then a penthouse. There's 2 floors that will be used for manufacturing, the top full-size and the ground. There'll be manufacturing, or excuse me, there'll be mechanical space that separates those 2 modules, and there'll be mechanical room on the top. This will be a fully licensed FDA regulated facility. It will also plan to be operated 24 hours a day, 7 days a week. So inside the facility, we'll have general manufacturing space, general warehouse space, we'll have administration space, and we'll also have biosafety level 1 and some biosafety level 2 space. The next slide. We do plan to use the U.S. Green Building Council LEED process to govern our design, and we're targeting certifying at a silver level. You can see this is a picture that's between the facilities, so you can see some of the sustainable aspects there where water retention is part of it. There'll be bike parking. The facility will be photovoltaic ready. We're not going to put photovoltaics on it as part of this project. That's really more of a site utility piece, but this will be ready to receive them. We'll comply with the natural gas ban in Berkeley, sorry. And it's also, we will have bird safe class. So, next slide. Let this one just rest a bit, and some dotted lines will show up that will help us discuss the setback. So you'll see some yellow lines appear. That is the volume requirements as part of our development agreement. You can see how this facility fits within those. It's set back from the property line further, and it's also, you can see how the roof height variations fit within those limits. Next slide. So just a little bit about what's going on inside the facility. You can see the facility there in the picture, and then you can see on the left-hand side of the slide, there's basically three boxes. Now I want to take a little bit of a step back here and talk about manufacturing. So we have 3 basic technologies that we'll be able to launch from. One is around protein therapeutics, which we have a cell culture technology center being built right now, and it's finishing up. There'll be a picture of it later. That's really around protein therapeutics, MAB technology, pretty standard right now in biotech. Then there will be a cell therapy process and a gene therapy process. As Jens mentioned, there's these 2 technologies. Many of the process unit operations are similar. However, how many you use and which ones you use to manufacture the specific product can vary. So what we do is we design general manufacturing spaces in a module, which allows us to build, so we know how big it's going to be, which allows us to build an open room and be comfortable that we can fit a module on each floor. So what you see here, the blue box on the top floor, the third floor, and the ground floor, those are manufacturing modules. We're going to outfit the third floor with a self-fit therapy module, and we'll wait for the demand on the ground floor. You can see a mechanical space in between, and there'll be a mechanical space above. Next slide. So this is just to give, now apologize for the eye chart, but you can see these are a little blow up of what would be in the modules. You can see a cell therapy module and a gene therapy module, they're the same size. You can see the way the rooms are arranged and the equipment is arranged is a bit different because there are a different product type. You can also see the type of space we need varies some. But just to give the group an idea. Next slide. Here's some pictures of the project, just moving clockwise, starting in the top left corner. That is taken from the railroad tracks looking towards Albany. You can see the existing Cell Culture Technology Center there, the shorter building. And then you can see the facility we're proposing there, the cell therapy and gene therapy production center. Moving to the right, that is between the buildings, looking back up towards Berkeley. Straight down, that's if you're on 4th Street looking south. You can see the cell therapy lies in the foreground. You can kind of see the CCTC in the background. And then finally in the bottom left corner there, that is looking between. You can see a little bit of the Cell Culture Technology Center, again, the existing facility on the left, and then the building where we're proposing on the right, and you're looking back towards the bay. Next slide. So to give everybody an idea of how you'll be able to see this from the edge of our campus, I believe this slide will advance and show. So this is looking south on 4th Street, and you can go back. This is looking south on 4th Street from Dwight. So between two existing facilities, let's see if it can do it. We have some fancy automation here. Wait for it. Yep, there it is. So that's the view when the building's built. You can't, so this basically shows us we can't see it from Dwight and 4th. Next slide. This is if you're at the end of Carlton. So Carlton runs into our site a bit along the McCully Foundry and stops about 5th Street. So if you were at the end where that road dead ends and looking onto site, you'd be able to see the facility there, and you see it next to the Cell Culture Technology Center. Next slide. We go to Aquatic Park. So if you're on Aquatic Park, that one flipped kind of fast. So Aquatic Park looking basically due east. On the path there in the park, looking east, you can see we had an older facility there that was a former data center. Technology advances allowed us not to need that anymore. And then when the new facility is there, slide, thank you. That's what you'll be able to see. Basically the top of it there. Next slide. So this is again in Aquatic Park, looking back towards Albany. So if you were south of the facility, looking northeast, you can see the facility through the trees there. Thank you very much for flipping this back and forth. Maybe one more time forward. There it is. So you can see it's in the shadow of the trees there. Next slide. A little bit about the landscaping. Pretty standard in Berkeley, all drought tolerant, mostly native. That includes some native plantings that are great with the plantings around it. Next slide. Some of the building finishes, it will be a metal panel facility with glass and have shades. A lot of that's driven through the green building process to allow for a low solar gain, but metal the building with an insulated panel, glass windows, and a flat roof, a high albedo. Next slide. So this is us looking west. I think this is pretty close to my last slide. So again, if you were looking basically due west from our campus, elevated stuff, this is what you'd see. The building on the right is our Cell Culture Technology Center that again is wrapping up. The building on the left, or excuse me, my bad. Left is the one that's finishing up. Right is the facility that we're proposing. One of the comments we did get from the Design Review Commission was the monotone of the facility, to maybe take a look at that and review that with staff for other options, which we are working on. Next slide. So that brings us to the end. So wanted to make sure we had plenty of time for questions, and also make sure we give a nice overview for everyone. So with questions, feel free to ask us to back up or elaborate. But with that, I'll stop my comments and I'll turn it back over to Kathy.
Thank you, Drew. So everyone, again, this is your time for asking questions, providing feedback on this new building. As indicated at the beginning of the program, we'd love to start with questions from the audience live. So feel free to raise your hand in the raise the hand symbol that you find on your screen. And then written questions, we'll get to those after about 15 minutes, and you can put those in the Q&A portion of the Zoom screen. If you just want to leave us a comment and you don't want to speak, feel free to put that in the chat. And with that, I'm going to start with Edward. Edward, do you want to take yourself off mute?
Will that do it?
Yes.
Yes, thank you very much. Very interesting. I live on 9th Street, just off Dwight Way, and I've been here for 30 some years now. And as far as I'm concerned, you guys have been great neighbors. I was involved in the West Berkeley project proposed by Mayor Bates maybe 10 or 11, 12 years ago. And I was opposed to what he was proposing to be done in West Berkeley regarding the expansion and height limit increases and those kinds of things. But my question, what I recall is that assuming you have 1,000 employees, my memory is that something like 5% of the people commuted by public transit, and that everybody else drove their own automobiles to the site. And if I'm curious, how many more employees are you planning to employ? And are you going to take any measures to ensure that the 6th Street and other streets here in West Berkeley don't become more jammed than they already are? Because there is, in fact, a tremendous amount of development that's just coming down the pike, and it's going to jam the streets up if we don't figure out some way to deal with the automobiles.
Edward, thank you for your question. So I think a bit of your question is about our amended our process underway to amend the development agreement. However, let me turn the mic over to Drew to speak a little bit to parking, traffic, and particularly associated with this building.
Yeah. So thanks for that. It's a great question, and I always appreciate being the engineer getting to answer it. So a couple of things, like Kathy mentioned, we are doing we're going through the process to extend our 30-year development agreement with the City of Berkeley. That is going through a full CEQA process, and the parking impacts and the needed mitigations will be addressed as part of that process. Speaking about this facility specifically, I don't know the total job count, but it's likely less than 100 folks. So less than a 10% add to site. Now, the other part of your question was traffic management and our incentives to get employees out of a single car. And that is clearly a focus. So a demand management plan is definitely part of our long-term. It's also part of we say we want to green the building. We want people to be able to get to it in a very efficient way as well. So I hope that answered your question.
Thank you. I'll keep optimistic.
Yes, me too.
Proof will be in the pudding, however.
Yes, agreed.
All right. I see no other hands up. Let me give a minute here and then I'll go to the written Q&A. Oh, and here we go. Alexandra, if you can unmute yourself.
Yes. Hi. Can you hear me?
Yeah, tell us
Hi.
if you'd like a little about yourself and ask us your question.
Yeah, I live and work very close to your facility. And I was interested in learning more about how Bayer is working to replace animal testing at this campus and whether there is animal testing happening and what kind of animals you have there.
Jens, do you want to field that question, please?
Sure, no, absolutely. So we don't actually have animals or any animal testing. And
Wonderful.
yes, and as a matter of fact, you know, when you think about it, I mean, from my personal perspective, will we, as humanity, developing therapies to both for people and animals, do we in the future still, as an industry, require animal tests? Probably. However, what I'm hoping personally is that you know, a lot of the technologies that we are working on right now, the cell therapies that we are developing, will more and more also allow us to use actually cells in bioreactors and cultures to basically simulate organ systems and then minimize over time more and more animal testing across the industry. So that's certainly a hope that a lot of the technologies that we are developing, because again, you know, when you think about it, what I said earlier, we are working on culturing heart cells in vitro so that you can then transplant them back into human to treat heart failure. Now, that basically means you have like real working human heart tissue in a reactor, which would be ideal then to replace an animal model to test other drugs in the future. So hopefully we can have a bit of a contribution there overall as well.
That's really wonderful. Thank you, Jens.
Sure.
Okay. Anyone else want to raise their hand? Oh, we got a couple more. Oh, and one I don't have a name on. Okay, so the person who is noted as 542450, could you unmute?
I don't know if that's me or not.
It is you.
Oh, okay, good. My name is Seema. Hello, everybody. It was a wonderful presentation. Thank you so much. And thank you for being a wonderful neighbor to us in Berkeley. I'm an architect and a lighting designer. So my question would be about the building and how you are considering energy saving in it, and I mean, more information about the LEED silver that you mentioned, and if this energy saving is covering some part of your lighting as well.
Drew, this sounds like a question just for you.
Yeah, so it's one of the things I really like about the LEED process is it has you model your facility that meets code, and then it drives you to make improvements. So overall energy efficiency that we achieve, I'm not totally sure what it'll be, but it should be more than 20% above code. The California Energy Code really kind of helps you here, because to build a building in California, it has to be fairly energy efficient as well. But as far as efficient lighting, absolutely. I will say that some of our processes are light sensitive, and also some of the work that we do, the looms on the desk are also important. So that can limit the, I would say, breadth of fixture that we use, but those are isolated areas. Elsewhere in the facility, highly efficient fixtures, motion sensors, active management of daylighting, and so on. All that will be deployed. As far as the exterior of the building, everything will be shielded, so we won't have any pollution off-site. But it's a great question. I hope I answered it.
Thank you so much. Yes, you did.
Thank you.
Jens, did you want to touch on anything further just about Bear sustainability objectives in general, just to give people context for why we do things this way?
Yeah, no, absolutely. I mean, this is really, really important to us as a company, as an organization of people that really, care about the environment. So and maybe just one thing to add, we also have globally the goal to be completely carbon neutral by 2030 worldwide. And so this entire topic is extremely important to us.
Thanks, Jens. Next, Denisha Delane. Introduce yourself, unmute, and tell us about what your question is.
Hi, good evening. I'm a Berkeley resident.
Oops.
Oh, what happened?
Oh, Denisha, we just lost you, but I think now you're back.
How about now? Can you hear me?
Yeah. Yes, thank you.
Awesome. So I'm over here on 6th Street and love you all as a neighbor, and am a old Berkeley High person that was able to see a number of young people go through the biotech program.
Great.
So my personal passion, also worked for the former council member in the area, Margaret Breland, who was heavily involved in a lot of old Bayer stuff. And so Edward mentioned a lot of things that were definitely questions that I was going to ask about traffic mitigation right at 6, because I live at the curve right at Dwight Wade Crescent. So if there are opportunities there to support some traffic mitigation, even with reduced cars, it's just a headache. And just kind of like a speedway there. It's not your problem alone, and it's not your fault, but it would be a nice contribution to aid in the neighborhood so that people aren't flying around that corner at over, crazy rates of speed. I did have a question though about, are there going to be any contributions or incentives to build pathways and strengthen those pathways with local high school students that are growing up in West Berkeley? How can we, how does the growth of the building also support some job training or internship opportunity for young people to kind of expose them to the field?
Oh, golly. Now, Denisha, you hit on a topic that's near and dear to every single person. And it'll be a battle for who gets to talk first.
I volunteer.
There you go, Jens.
I would love to. No, Denisha, this is a really important topic for us. And just to give you a little bit of background, so when I came here in January 2000, so I signed up in '99, came over in January 2000. And I actually mentored, working very closely with biotech partners, and I mentored personally also high school students here from Berkeley, and then also community college students. This is something that is really important, and where I feel that we can play a very important role, because again, we are a manufacturing site. I mean, high tech, yes, but it's not research. It's development and manufacturing in particular, which means that we have a whole variety of jobs. And what we want to do is we want to expand the talent pool even more, and we want to help young kids, show them a path that they can actually contribute to society by having jobs working side by side with us on making these important medicines. And so a lot of our folks at the site come from the local community and have gone through these programs. And at least one of the students that I mentored is also still now 20 years later, now of course in a much more senior role in the organization, but part of the Bayer team. So this is something that we want to build on and that we want to expand. And we are hoping that we can over time create quite a few additional jobs, as Drew also pointed out, and opportunities for people in the community.
Awesome. Oh, I forgot to add, this is more for Drew. Drew, so of course, I almost used to set my morning alarm by the alarms at Bayer. Will there be any new alarms coming? They're not problematic. When I moved in here over 10 years ago, I was like, what is that? Now I'm pretty used to them. Are there any new alarms coming?
No new alarms, and we're actually working on our Wednesday at noon, so it'll be less annoying and more understandable. And the horns that you hear a little randomly, those are driven by problems that we're working on every single day. So hopefully those will reduce. And they
Gotcha.
have. Thanks for the question, though. Nice to know
Thanks.
Thanks, Denisha. Okay, I am gonna go over to the written Q&A and get started there. So the first couple of questions are from Shanti Baker. Shanti, thank you for submitting your questions. I'm going to hit one of them pretty quickly, and then I'm going to ask Jens to answer the second one. So your first question is about biosafety implications of the new facility and how does it add to Bayer's existing situation. So on site today, we're biosafety level 1 and 2. The new building, biosafety level 1 and 2. And just to clarify for folks, because as a layperson myself, not a scientist, I always ask the question, well, what does that mean? And biosafety level 1 and 2 is the same level that you would find in the laboratory at your local hospital, essentially. So that's the biosafety levels we operate at. Now, in terms of your question about prevention of disease, I'm going to turn it over to the scientists and ask Jens to give an answer.
Sure. Yeah. So we are doing quite a bit. I mean, first of all, we are supporting health and wellness programs actually globally in all the communities that we have site and activities in, and of course, you know, as you heard earlier, our impact on the environment is, you know, it's very important for us that we minimize the impact on the environment and actually build sustainably, and which also helps ultimately all across board. And we also you know, have a variety of products that support prevention, disease prevention. I mean, if you look at aspirin cardio, for example, which really helps prevent cardiovascular disease and heart attack. When you look at, I mean, even, you know, we do have vitamin products and supplements that help you, I mean, help you to keep healthy. But the fact is, and the very unfortunate fact is, that no matter how much you try to prevent disease, many diseases are just not preventable. And you know, I mean, I had a good friend of mine, one of my closest friends, pass away recently of pancreatic cancer, and I can tell you, I mean, he was a health fanatic, and he did everything you possibly could to prevent disease, and still, unfortunately, he died because of cancer. So that's why, you know, beyond just prevention, we want to help treat these diseases that are just not preventable.
Thanks, Jens. So the next question, Jens, is from Michelle Nimitz, and she caught on that you talked about the revolutionizing of medicine by cell and gene therapies, and has asked if you could speak a little bit more about how this is different from current patient care and why you think it's revolutionary.
No, absolutely. And you know, when you look at medicines, a lot of times we are left with treating symptoms with traditional pharmaceuticals, especially small molecules, chemical drugs. A lot of times we are left with treating symptoms maybe delaying the progress of disease. If you look at cancer therapy, for example, I've been in this industry for a long time, and like I said, I mean, I went into pharma to really help people and to treat people and to save lives. And over many years, I've been really frustrated by, like in the cancer field, the fact that it's so hard to treat that despite our best efforts, some of the conventional therapies are really only sometimes extending life by a couple of months. Now, just, for people that are actually affected, just a couple months more with their families means a lot. But what we are working on now is taking it to the next level. When you look at cell therapy for cancer, as I mentioned earlier, the first ones are for hematological cancers. You get these response rates where like 94% in some cases of patients respond, have a complete response actually in some cases, and are healthy even years later. So basically you see the cancer melt away. And so that is a whole other paradigm now. The other thing is, again, in the area of gene therapy, where you could potentially really also cure these diseases like hemophilia and others, rather than what success meant previously, turning it into sort of a chronic ailment that you can keep in check. You could really, for the first time, think about curing people. And that indeed, if we are successful with that, and we are optimistic because, again, there's already so much progress happening, that is indeed nothing short of revolutionary.
Thank you, Jens. So Jennifer, I'm going to ask if you could please bring up the timeline slide again from the deck. There's been a question if we could walk through that again. So Drew, this is going to you to just walk through that again. And I think there are a couple of other questions associated with timeline that are in the Q&A. Asking about what it looks like past the meetings, when would construction start, how are we selecting general contractors, has an architect been selected? Yeah. So all of those at once, good for you.
Very good. So this is, again, the submission timeline. We started back in August, and we're here today on the 4th. Past this, so our zoning adjustment boards meeting would be our last, and assuming approval there, we would move to City Council in April, and then receive a use permit at that point. At that point, we would start construction as soon as possible after that, likely as early as April or May. The facility will look like a facility from the outside, starting in the end of the year, beginning of next year. And then we'll start the outfit of the actual production module on the inside for cell therapy. They're moving in parallel paths. So, construction that you can see through about the next year, then a lot of installation on the inside of the facility, wrapping up in probably late 2022, startup of the facility in early 2023, and go into production in the second half of 2023. That's all to support these products for our patients. Jens mentioned it, that and I kind of skipped over it. From an engineering perspective, this infrastructure is very different than infrastructure that we had in the past. In drug manufacturing and in biotech manufacturing, facilities were built around a larger patient population, and medicine was manufactured that could treat a broad population with a certain level of effects, say 60-65% would be effective. These new therapies, our ability to understand the patient and the condition and target the medicine at those two is elevated. And what that means is that our medicines are more targeted, which means inherently they're a little smaller population, but there's more of them. So these facilities have to be far more flexible in the way they're built and operated. So they're also hope everybody takes away from this that this is new tech, and we are investing heavily in this so we can meet patients as soon as possible. In days past, from the time the product got into the clinic, the time it could be to the patients could be many, many years. We hope to get that down to just a couple. And that is extremely important. One of the reasons why we're advancing the shell and then investing in that module. Now, I might have missed some of the question in there. Has an architect been picked? The answer is yes, there's two actually. So Fluor, who is a large engineering company, their biotech division is our main contractor, as well as they designed the shell. The interior module part of the facility, that's being designed by an Irish company called PM Group. They have a fairly large operation here in the Bay Area, headquartered in Oakland, actually. And their board member lives in Albany. So I like to call them a local company with an Irish accent, but they do have a fairly large here, but they operate globally. Kathy, did I miss a question in there? I kind of got off on my soapbox, but I love that part, so I wanted to talk about it.
So just a couple clarifiers. First, because you and I both are talking all the time about this project and the DA, but this project, the final approval is at Zoning and Justice Board. So I just want to clarify that for everyone. And then there was, I think you hit on it, but just to be sure, general contractor for this project has also been selected?
Yeah, that's Fluor.
Okay, just want to make sure we hit all those terms.
Yes.
And while we're talking about construction, and Jennifer, you can take the timeline down so everybody can see Drew and Jens big. Miguel has asked a question, and I'm going to read it out loud, Drew, and then I'd like you to answer. So Drew Miguel is a journeyman at a local 378 and a West Berkeley resident. There are about 5 iron workers in his union who are Berkeley residents. Are we hiring local residents as a requirement for our construction development? Apparently he knows that Oakland has some sort of local hiring requirements for contractors doing work.
So let me answer it 2 ways. We do not require local. Most of our contracting is done through our union trade contractors who so like Fluor is a union contractor. We do hold it a little bit merit because sometimes there's a real special piece of technology that's not covered by a local, but for the most part, 95 to 99% is done by local journeymen. As far as local, necessary requirement are the subcontractors we hire. They usually pull from the halls that are local. I hope that answers your question. I have to admit I'm a step away from the labor rules for each of the different unions on how they put labor onto projects, whether it's a open solicitation or a seniority or some other basis. We typically just follow the hall.
But Drew, a large percentage of our work for construction projects is done by union labor, correct?
Absolutely. It's, I mean, dollar wise, it's probably pushing 99%. From a labor standpoint, it's up in that per percentage too. Get the question in the past, why not 100? The space between 99.9 and 100 happens to be high. We do deal with some very special equipment that has some very special skills to adjust it and kind of finish it. And we just need to make sure that we have access to that quickly. So.
Super. So the next question on the list is from Sarah. And she's asking, out of curiosity, how sensitive is our cell and gene research, which I'm going to modify and say development and manufacturing because we don't do basic research on site. to light, because she noticed that we have small windows planned. Jens, do you want to take that one?
Actually, if you don't mind, I hand this over to Drew, because I don't think that this is necessarily due to the cells themselves.
Yeah, so we do have some media components and other things that we handle in our process that are sensitive to light. We arrange our interior to make sure that they're properly shielded, and we also keep them in equipment that can help shield that as well. The windows on the building, this is a very constant debate. They're expensive, and they also have to be maintained. And the way our equipment is arranged on the inside, they can kind of be lost behind things. So we arrange them that way. So the glass in the facility is really driven by kind of the arrangement of the inside, and also managing our costs.
And maybe to add, you know, I've worked in various different buildings before. You certainly have to also regulate the temperature very much in these kind of facilities. And so you can imagine that while the cells themselves are not sensitive to lights, yes, other components are, but you know, one big effect if you have big windows on a big facility like this is that it's going to heat up like crazy, and then you basically have to run your air conditioning at higher, so you increase energy consumption, and that's bad for the environment. So it's sort of a compromise.
Super. I just was checking the chat just to make sure there was nothing over there, and I caught one thing really quick. There is a question that ended up over in the chat about as this facility is going to be designed, of course, to meet FDA validation standards for manufacturing. Will you be commissioning it to achieve BMBL commissioning requirements for those areas operating as BSL 1 or 2? Drew, do you know the answer to this question? I'm not sure what BMBL is.
Yeah, so maybe I can have Scott Probst unmute here in a second. So we commission and qualify our facilities to an ISO class 7 or class 8. We also do the standard commissioning and qualification in our BSL levels. I don't know much about BMBL, but I do know that the requirements are significantly lower than what we go to, but maybe Scott can explain the details and tell me where I was wrong.
And for everybody to know, Scott Probst is another member of our scientific team on site, and he's the product lead for the Parkinson's cell therapy.
On mute.
You're on mute.
Oh,
Yeah.
or the headphone is not plugged in.
Right now, Scott.
You're not on mute now, Scott.
But somehow the microphone is not working.
Okay. How about if, well, Scott, how did I do? Thumbs up?
Now he might not be hearing you either.
Now he might not be able to hear you either. Okay,
I think Drew, you were spot on. I think the key is that the requirements that we are following we are actually exceeding the BMBL, not just meeting them.
And you know, if we have further clarification on that answer, we'll certainly follow up directly via email. I always love these questions. Michael Gomez has asked, when do you expect to be hiring new team members for this facility? Jens or Drew, I'm not sure which one of you would like to answer that, because I know everybody's excited about that.
I can answer, check the job board. I mean, these construction projects allow us to build the team and allow them to understand the manufacturing process, as well as the engineering support that's required and the testing support. So the teams build slow over, I would say, about the 2.5 years, but we are actively recruiting now for the initial positions.
Right. Right. And I think just a plug for those who are interested in jobs at Bear. There's a wide variety of jobs that you can always find at Bear. And we love having folks apply. So please consider looking at career.bear.us and taking a look at the opportunities available in Berkeley. So I don't see any more questions in the written queue. And no one has done a late breaking hands up, but I'm going to give it a minute or two if anybody wants to raise their hand for a question or a comment or put anything in the Q&A box. Going to give it a minute. And then Jens, I'll ask you to wrap it up.